Thank you, Chun, for the nice introduction. Thank you to the program chairs for inviting me to speak today. I'm really excited to talk to you about current and future integrations of genomics and AI. And I do want to point out that this presentation, the first half is on things that we know we can do, and the latter half is on things that I expect us to be able to do in the near future. And I think that one of the reasons I'm invited today is just because AI has become such a big part of our lives, and it's emerging over time. And it's amazing that we've gone from very poor models for things like self-driving to newer models that tech companies are developing that can take just vision pixels from the external world and allow cars to drive. Sorry, are you able to hear me better now? Allow cars to drive on their own. And this is getting better and better, and I think we'll see improvements in this over time to the point where it'll be unusual for us to drive ourselves. Perhaps a more relevant example to our bioinformatics colleagues here is using ChatGPT type models to assist us with our coding challenges. So I'm a native R scripter, but with a chatbot, I can easily write Python code. I can write shell scripts just by prompting this AI chatbot. And probably even more common in this audience is when you're asked to make a presentation that you can ask for, I'd like to make an image. And you can say, oh, I'd like to make an image more complex. And so I can present to you a summary of my presentation here. Of course, I don't want to go and it's not that complex. I want to bring this talk to a general non-AI audience here. So we're going to talk about AI data structure. We're talking about training techniques. And as I mentioned before, we'll talk about current implementations, things that we know work, and then I'll speculate on things that I think will work in the future. So how do we define artificial intelligence? To be honest, I used to really dislike the term artificial intelligence, especially just a few years ago when we were using mostly statistical models to do classification tasks. But with the advent of self-supervised learning algorithms, things that can learn from large sets of data, we're really approaching a situation where artificial intelligence can do superhuman tasks. And I think that as these new techniques emerge, we can really see the power of these AI, like true artificial intelligence models. So one of the elegant aspects of AI is how simple the data structure is. So all the data that's used for AI is essentially arrangement of numbers. So on the far left, there's a vector. It's just a one-dimensional array of numbers. And then matrix is two-dimensional. And then a tensor is 3 or more dimensions. And this is really the substrate of all artificial intelligence. Of course, within genomics, we already have our data structures. We have FASTQ files, we have BAM files, we have VCF files. And when my boss came to me and said, you know, you're doing this artificial intelligence stuff with images, we have lots of genomic data at Memorial, why can't we build artificial intelligence models? And I was actually really resistant to this because bioinformatics data structure is so organized, and I always thought of artificial intelligence being more useful in situations where we don't have structure, things like text. Text is largely unstructured. With AI, we can make it into more structured data. With medical images, we don't really have a foundational structure, so we use artificial intelligence. I've only recently come around to the fact that in the setting of highly structured data, like bioinformatics data, we can actually perform really high-quality artificial intelligence. And we do that by converting the data into these AI data structures. So taking a BAM file to a tensor, essentially. So the power of AI is in being able to work with all different types of data. So you can take text data, things from the internet, patient reports, EMR data, take that text data and feed it into tensor architecture and train GPT-like models. In the same sense, you can take medical images, convert them into a tensor data structure, and learn from medical images. What I'm proposing today is that we can do similar things with DNA, and I'll show you how we've done that in the past and how we're going to do it in the future. So thinking about learning representations, or feature representations in AI. So I'm breaking this presentation down into 4 different aspects. So an unsupervised approach and supervised approach, which I think most people in the room will be familiar with. And then at the end of the lecture, I'll talk about the more challenging but more exciting ways to use artificial intelligence. So unsupervised learning has been around for a really long time. I'm sure everybody has created a PCA plot at some point in their lives, where you take something that is higher dimension, maybe 100 features or more, and you can condense that data into its principal components, and then you can plot that in a 2D graph. More recent advancements are TSNE and UMAP, which basically take large amounts of data, and then it finds relationships between the data and pushes the things together that are similar and the things that are different farther apart. And then UMAP is kind of a updated implementation of TSNE. So in the situation of genomics data, we have really large data sets, things like methylation arrays, which typically come in 450,000 to 750,000 features. We can take these really large data structures, say 450,000 data points. We can compress for each sample that information down into a single point on a Cartesian plot in 2 dimensions or 3 dimensions. And to illustrate this is possible, and I'm not the first person to do this, but I invite you to scan the QR code if you're able to. And I shared on my GitHub a nice example of how a TSNE can be used with genomics data. And this is taking public data. It's taking TCGA methylation data, as well as all of the data that's been available through Heidelberg in sarcoma and glioma. And I generated this plot, and you can explore it on your phone. It's better on the computer because you're able to zoom in better, but I'll invite you to do that. I'll just take a little tour of this data. It's a video, so I'll try to do a play-by-play. So you can see this is a 3-dimensional representation of 13,000 individual tumors, and you can toggle the plot. You can see that there's several different clouds. I left the labels on just so we can see more clearly what the tumor types are, hopefully, although it's a little bit hard. So this here is the sarcoma cluster, and there's a bunch of different sarcoma types, endometrial sarcomas, and you can see the different colors clustered together. So there's a cluster of leiomyosarcoma in here. As we move over here, we'll see rhabdoid tumors that are clustering. There's a rare tumors. They cluster very nicely. Then we have angiosarcoma. Okay, so now we're going to move over to a different cluster. This is the glioma cluster, which is mostly Heidelberg data, but there's also TCGA. It's a good proof of principle that tumors that come from Heidelberg and tumors that come from the TCGA, if they're gliomas, they cluster together in the same spot. We're going to move over to the largest cluster here in a second. Sorry, I can't control the speed of the video. It's a little bit hard to navigate. So here we're moving to the carcinomas. There's a bunch of breast cancers, a bunch of thyroid cancers. And we're zooming back out. Next step, we're gonna move through a big cloud of melanomas, which is a mix of TCGA and Heidelberg data. And then beyond that, we'll see another cluster that's mostly squamous cell carcinomas. And I just wanted to point out one thing. So in this squamous cell carcinoma cluster, there's lung squamous cell carcinoma, there's head and neck squamous cell carcinoma, there's esophageal squamous cell carcinoma, which they're all kind of related as to where they come from. There's also an area of bladder cancer over here. And then we're moving along through the different GI carcinomas. And then on the other side of this, there's another set of squamous cell carcinomas that are mostly cervical cancer, so they're mostly probably HPV-associated cancers. And then there's another set over here that's bladder cancer. So there's probably some difference between the bladder cancer that clusters over with HPV-related squamous cell carcinomas and the ones that cluster with the other squamous cell carcinomas. So feel free to play around with this. I thought it's pretty cool. Now, the unsupervised clustering is interesting. It's fun to look at. It's nice for presentations like this. It's not really useful in any clinical sense. It's hard to describe like where on a three-dimensional matrix a specific cancer lives. So, but on the other hand, it does tell us whether there's learnable features related to different things like this methylation data. So once you've shown that there's learnable data, then you can train a supervised model. And this has been done pretty extensively. The most famous group, I think, was mentioned in the previous talk by one of the questions. There's a group out of Germany in Heidelberg that has identified a huge subset of neuropathology tumors, mostly gliomas, and they found that it's actually more straightforward to define the specific types of gliomas according to their methylation pattern. And they trained a model, a machine learning model, mostly random forest. They call it an ensemble model, but it's mostly random forest. And they show that you can accurately predict the tumor type based on the methylation array. And this is such a powerful model. It's just a general model that we actually use it in clinical care. At MSK, we actually offer this as a test for our neuropathology and neurosurgery patients. We often get a lot of consults on very complex neuropathology cases, so we'll run the methylation array, and then we'll output a very specific tumor type, and it's very useful in clinical practice. It's one of the few widely recognized uses of machine learning currently used in clinical practice. So I want to introduce a new way of doing classification using more advanced AI techniques or using convolutional neural networks. So the methylation data comes in an array. What I decided to do is to convert the 400,000 CPG site array into a three-dimensional tensor. So instead of having this super long tensor that from one end to the other, it's 400,000 units away. By converting to a tensor, you compact that data into a 200 by 200 by 10 tensor that contains the same amount of data as the long array. And by doing this, you create an architecture that's very optimized for doing modern AI. So it works well. Once you have a tensor, you can run PyTorch libraries, which is the kind of de facto Python package for doing machine learning models. And this data structure is also optimized for working with GPUs. So you can train a machine learning model very quickly. It only takes a few minutes to do what I'm going to show you next. So I took the 8,000 tumors from the TCGA that have methylation data, and I trained a model to predict the tumor type. The AUC is, for most tumors, it's 1. But for the lowest tumor, it was 0.989. And you can see if I can use this. You can see there's the diagonal line basically shows the samples that agree with the actual. So the x-axis is the predicted tumor type, and the y-axis is the true type. And the only errors we see are the kind of predictable errors, because TCGA happens to have rectal, which is separated from colon cancer, even though they're pretty much the same cancer. So this wasn't super surprising, but what was surprising to me is you can actually create tensors out of RNA-seq data and get similar performance with TCGA. I've also done the Heidelberg general tumor glioma classifier. I've done the very detailed 81 classifier that they reported in using random forest, and the performance is at least on par with what the Heidelberg group showed. I also did sarcoma. I think that this is actually quite a bit better for sarcoma, but I haven't published this yet. So these are things that we know we can do with AI. You can train supervised models where you have very informative data. What we haven't yet quite figured out yet is how do we use next generation sequencing data? How can we apply machine learning or artificial intelligence in next generation sequencing? So relative to methylation data, NGS is much less structured. We use different panels in different institutions. There's different sequencers that we use. We also use NGS for lots of different things, whereas with like RNA-seq, we just look for whole transcriptome for the most part. But in NGS on DNA, we'll look for point mutations, indels, copy numbers, structural variants. You can also do genome-wide predictions like microsatellite instability or homologous repair deficiency. So how can we use AI for this? And the challenge relative to methylation data is just how large the data is. So the methylation array is on the order of 400,000 parameters. For whole exome sequencing, we're looking at more like 8 billion parameters. So it's many orders of magnitude scale different from our array-based data. Fortunately, we have a little bit of a experience with working with large data sets, and that's actually the focus of my research, my funded research lab, is working with whole slide images. So whole slide images, probably many people in here are familiar with them. They're enormous in size. So this here is maybe like a 600 by 600 pixel image of a lung cancer. And the slide is labeled as being positive or negative for whatever biomarker, in this case CGFR. In order to actually process this data with AI models, we have to sub-segment the slide down into small patches. And it's a little bit hard to see, but this is basically a grid that shows all the individual patches. And they look very small on the big image, but they're actually relatively large on the big image. In fact, the 224 by 224 pixel patches, the size of ImageNet images, which is kind of the standard for learning from images with AI. And there's on the order of 10,000 to 100,000 individual patches per whole slide image. So that actually puts whole slide images on the same order of magnitude as the amount of data that we see in whole exome sequencing. So given that they're similar data sizes, can we use what we've learned from working with whole slide images and apply that same technique to next generation sequencing? The principal challenge of having really large data sets is you can't fit the entire whole slide image or whole exome sequencing BAM file onto a single GPU. And also, the types of things we want to learn from these cases, NGS or whole slide images, are usually only labeled at the whole level, and we don't know exactly which part of the exome, for example, is responsible for things like homologous repair deficiency, which is the same problem we have with the whole slide level. We don't know what part of the slide tells us whether it's an EGFR positive cancer or not. So the way that we've done this with whole slide images is something called weakly supervised learning. It's more popularly known as multiple instance learning, or at least that's the most common technique. And in weakly supervised learning, we take patches out of the whole slide image, like I mentioned before, and these are 224 by 224 pixel patches. They're color images, so there's a red, green, blue channel. So you basically have 3 numbers per unit. So it's 224 by 224 by 3 is the size of our data. We convert that to a tensor, straightforward process. And then in order to actually process all the information from each individual patch, we have to pass that data through a feature extraction encoder, which I call a pre-trained encoder here. And what that pre-trained encoder does is it takes the salient features from that image and reduces the size of the data down from 224 by 224 by 3 down to on the order of 1,000 units. So now you have 1,000 unit vector for every patch on the whole slide image. And that becomes a manageable amount of data that you can train a model on the whole slide image using something we call an aggregation function. So then you can predict the slide level probability for whatever biomarker your AI model is being trained on. So the pre-trained encoder is the critical unit in this architecture. And you may hear the term foundation model or AI foundation model. In text, GPT is an AI foundation model. In pathology, we just call them whole slide image foundation models. So these models are trained in what we call a self-supervised manner, where you take a known image, this is a flamingo, and you extract portions of the flamingo, and you hold out other portions, and the model has to learn using an encoder what the features are that are likely to be in these missing patches, and then it decodes it back to the original image, and the difference between the target image, or the actual image and what's predicted, can be learned. I have another slide. So this is a better example. So here you have a fox. You mask most of the image. The model tries to reconstruct it. This is real. This is from a Facebook model called Mass Autoencoder. And then the image is reconstructed here, and then whatever the difference between the reconstruction and the original is the loss. And the AI model tries to minimize that loss over time. This is exactly how GPTs are done. So this is just a sentence. You mask out some parts of the sentence. The model tries to predict, and whatever is different between the predicted part and the original is the loss, and that's learned over time. So the advantage of these models, which they call this autoregressive transformers or self-supervised learning, is that it takes completely unlabeled data, and you can scale this infinitely. So as long as you can find images, as long as you can find text, you can continue to improve this. And the larger the model you form, the more data you expose to it, the better they perform. And this is proven out very nicely with whole slide images. There's been 3 major foundation models reported in the last few months for whole slide image encoders, and these are all open source now, so we use them in our lab. So then the question is, can we use this architecture for genomics foundation models? And I propose that we can. So all I did here was I switched out the image to data coming off the sequencer, so mostly whole exome sequencing. You generate BAM files, you convert the BAM files to tensors, you train the model, et cetera. And then in order to build the foundation model, you extract all the regions of the genome and convert those to tensors, and then you can do some kind of self-supervised learning. I would probably use something similar to what we use for all of the whole slide image foundation models. And we actually have a kind of older publication. This is from Memorial. One of my friends, John Ziegler, came up with this idea where he used the AI at the time to predict microsatellite instability. And this is actually going to come out as a publication in the next few months. So what he did is he wrote a script that will take a BAM file, convert the BAM file into tensors. These regions of the genome were selected as microsatellites in the same way we do for MSI sensor. So we have vector representations of regions of the genome that contain the microsatellites. And he trained an encoder, which he actually doesn't have on this figure. He trained an encoder that will learn from whether a site is a microsatellite or not, and pull the higher data size down to a smaller vector size, and then you aggregate all the microsatellites across the genome, and it allows you to predict the microsatellite instability status. We actually use this as a sort of a backup technique in our next generation sequencing platform. So we use MSI sensor as our main MSI caller, but this model is actually better when there's lower tumor fraction and other things. But the reason I bring it to your attention is the architecture is almost exactly the same as the multiple instance learning that I showed earlier, the weakly supervised learning. The only difference is that we don't have a foundation model trained yet on whole exome data. So we can use whole exome data from TCGA or in-house data, train a foundation model in theory, and we can then use that as a feature extraction encoder to train whole genome level models. So we don't know whether this is going to work, but based on what we've seen For images, we know that we can learn salient features from images. And since the data architecture is the same, and we have on the same scale of data as we do for whole slide images, we have thousands of examples. We have the entire TCGA whole exome sequencing. We have our internal cohort at MSK. So I think it could be an effective way of using AI in genomics. The types of tasks that I would be interested in, we've kind of proved that microsatellite instability can be learned. What about homologous repair deficiency? There's already some methods for using NGS to predict homologous repair deficiency, and we know the mechanism, but perhaps we can train a more robust model for predicting HRD. You can also start to think about things that are where we have no idea what the features might be, like whether a patient responds to a specific chemotherapy or not. We wouldn't know even where to look in the genome to predict that for some types of chemotherapy. And we can also think about survival. So I hope in this talk that I've given you an idea of how AI can be used in genomics. I hope I've shown you the current ways that we can do these things, and I think that we have an interesting future here. So I hope in maybe a year or two you can invite me back and I can show you the results once we've trained a foundation model in genomics. So that ends my talk. Here's my acknowledgments, including my grant funding and my AI scientists in my lab. And I'm happy to answer questions in the next 2 minutes and 43 seconds.
Hi, great talk. I just want to make a comment that the use of AI tool in genomics has great potential, because if you think about the application of it, it can be used as a screening tool for NGS, what is the pretest probability of having a positive finding, let's say MSI, before you do it. If the pretest probability is 10, very low, you might not be worth paying, $2,000. So, I mean, the tool is really help to screen and triage and making better sense of doing expensive molecular testing.
Yeah, especially if you can do it on just a whole slide image. The cost of scanning an image is quite low. That's actually the focus of my research laboratory. We have a model that predicts EGFR almost perfectly. And because EGFR is such a critical biomarker in lung cancer and the desire for turnaround time is so fast, if we can give them the results of an AI model within like a few hours of when the tissue is processed, then we can triage the tissue. We can do a rapid EGFR test if we want to using Adilla, or if the model believes that the model predicts that EGFR is less likely, then you can prioritize the tissue for more broad genomic profiling. Okay.
Hi, really exciting work. I was wondering if we could hear your thoughts about how AI like this might compare to like haplotype imputation for whole genome sequencing, what kind of improvements this might present?
I mean, I'll just answer in a general sense. I don't think that AI is the answer to everything, but I think that if you can think of a way to, and I'm not super familiar with that technique, sorry, but if you can think of a way to structure your data such that you have a lot of training data, then you might be able to improve on whatever the existing models are. But I also think that if you don't need something super sophisticated like a foundation model, it's a lot, and you can use something more straightforward like a random forest model, then you probably want to do that.
Yeah, gotcha. Thank you.
Sorry, unfortunately, we don't have the time for the.
I think so. Is it working?
Please feel free to stop chat anytime if you have more questions.
Yeah, I think there's a roundtable at the end of the day where we can talk about things. So thank you.